Hi, my name is Barsha Garish, and I live in Wexford, Pennsylvania. And my career goal is to become an anesthesiologist, and the knowledge of microbiology will help that because it will help prevent complications during when a patient is undergoing anesthesia. And my title was to determine which soil contains bacteria that interacts with sun hemp legumes in Wexford, Pennsylvania. And my hypothesis was that the soil from the positive control would interact best with the legume seeds and have the most germination and nodules. And the methodology behind this research was basically to collect soil from two different locations. And for the third pot, I used soil from location one and I added an inoculant, which served as the positive control. And I observed these plants for 2 to 3 weeks to observe germination. And after 3 weeks, I took out two plants from each pot to look for nodules in the roots. And there weren't any nodules on the positive control, but I did find nodules for the plants in the front yard and backyard. And I observed those nodules under the microscope. And I put them back in, and at the 3.5 mark, I took out the nodules from the front and backyard. And at this time, the positive control still didn't have any nodules. So I took the nodules from the front and backyard plants and I created a mixture using the nodules, which I then spread on agar plates to observe for colony growth. And I couldn't do this for the positive control until 4 weeks when the plant finally had nodules. Here you can see the images for colonies on the positive control, the front yard, and the backyard. And you can see that they're all different shapes and sizes. Here they're very small, circular shaped, and there's a large number of them, whereas over here there's not as many of them and they're very different shapes. Some of them are small, some of them are big, and here it's like a mixture of the two colonies from positive control and the backyard, whereas a lot of them are small but then some of them are big, and you can kind of see that it looks like some from the positive control and some from the backyard. And here I put a graph of the plant height, and you can see that the positive control had the greatest height over the 4 week span, whereas the front yard and backyard had heights varying from 20 to 25 centimeters. Here I included the images of the separate plants from each pot, and I did the exact same labels. So this plant was from the positive control, this plant was from the front yard, and this plant was from the backyard. And here in the microscope images, this one was from the front yard and this one was from the backyard. And the front yard, the nodules over here looked a little pink, and that indicates that nitrogen fixation has occurred. Whereas over here, the nodules were white, meaning that nitrogen fixation has not occurred. And from these results, I concluded that the backyard soil interacted best with the legume seeds and the bacteria, whereas the positive control interacted the poorest with the legume seeds and the bacteria, which disproves my hypothesis because I said the positive control would interact best, which was incorrect. And lastly, I would like to acknowledge Jetty for assisting me through this process, and to Nicole for her knowledge in the college admission process. And I'd also like to thank Moon Prep and UMass for the opportunity. And are there any questions?